thanks for inviting me to give this talk and tell you about what's going on in our research group. So I think the place I want to start is by saying that, I'm sure most of you know that there's a lot of research going on focused on autism here at the Mind Institute. And there's also a lot of research going on focused on Fragile X syndrome. But in some ways, those two topics kind of run in parallel. And there's a lot of in-depth investigation going on within each category, but I think there's a lot of reasons to look at the overlap between the two and how studying one can inform our understanding of the other. So that's kind of the theme of the talk. And the way that we're going to work this is I'm going to kind of introduce the topic and tell you a little bit about Fragile X syndrome. And then Lisa is going to tell you a bit more about some overlapping ideas, symptoms, overlapping ideas pertaining to autism, anxiety symptoms, and Fragile X. And you'll see what I mean when she goes over her material. So let's see. Let me start by giving you a quick overview of Fragile X. I think most people in the room know quite a bit about it, but there may be some people here or watching through the Ethernet who haven't heard of it. It's the most common inherited cause of intellectual disability, and it occurs in somewhere between 1 in 2,500 and 1 in 5,000 live births. It's caused by an expansion or a mutation on a gene on the X chromosome. The gene's called FMR1. And this gene, within this gene, there's a repeating CGG. And in normal individuals, it repeats somewhere between about 5 and 40-ish times. In individuals with the full mutation, which I'm showing here on the bottom, this expansion gets very large and goes over 200, and then when that happens, there's a process called methylation, which I won't get into today, but basically that process just shuts the gene off and then it stops making a protein called FMRP, Fragile X Mental Retardation 1 protein. And when that protein is absent or reduced, then there's changes in the brain that occur that lead to the cognitive impairments and behavioral abnormalities that are characteristic, which I'll talk about in a minute. I'm not going to spend too much time talking about the premutation, which is where an individual has between 55 and 200 of these repeats. And that gives rise to at least a couple of other totally separate conditions called Fragile X associated tremor ataxia syndrome and premature ovarian insufficiency, and that's in only women, obviously. So we're going to focus mainly on Fragile X. So in terms of this overlap that I mentioned, we know from epidemiological studies that somewhere between about 3 and 5% of all individuals with autism have their autism due to the Fragile X mutation. And on the other side of the coin, about 30 to 50% of individuals with Fragile X, and I put in parentheses males because of some specific differences in the way that the gene functions in females, but in males who are usually much more affected, there's a percentage estimate of about 30 to 50% that meet diagnostic criteria for autism. So the symptoms that these individuals with Fragile X and autism have are in many ways and in most cases very similar, if not completely overlapping with autism that you see, or autistic symptoms you see in idiopathic autism. So in the communication domain, you see echolalia, repetitive speech, difficulty with conversation, and so on. In terms of reciprocal social interaction, we see poor eye contact, lack of peer relationships and avoidance, problems with joint attention, lack of pointing, many of the classic things that you see in people with regular or idiopathic autism. Individuals with Fragile X, even if they don't have full autism, they often have lots of stereotypic behaviors like hand flapping or obsessive compulsive behaviors, intense interests, and so on. And then sensory abnormalities are not considered to be part of the diagnostic criteria for autism, but it so happens that individuals with Fragile X who have autism and individuals with idiopathic autism also have many of the same sensory either hypersensitivities or hyposensitivities. So I wanted to throw in a slide also to bring up this controversy that I think is waning now in the field, but where some individuals feel that autism in somebody with Fragile X is not real autism. And so I think I could spend a whole other hour talking about this controversy, but I think one question that we could ask in response to that is, what is real autism? And the answer is we don't really know, because we're learning now there's so many different etiologies or causes, so many different pathways for a person to reach an endophenotype or a presentation of autistic disorder that we basically are now recognizing that Fragile X is just one of those many etiologies. So the fact is that although there might be still some controversy about this, many individuals who have Fragile X syndrome meet criteria for autism according to DSM-4 criteria, the ADOS, which is a semi-structured interaction that's part of the gold standard for evaluating autism, and the ADI, which is the Autism Diagnostic Interview, which is done with parents. So our understanding then, like I mentioned before, is that Fragile X autism is just really one of the autisms. And so the premise that comes from this is that Fragile X is a useful and more homogeneous or more kind of circumscribed model condition for understanding autism symptoms and behaviors. So within our field, Fragile X is often described as a disorder that can really tell us something about the pathophysiology of autism. Let's see, I think I was supposed to not advance that slide because Lisa is next. Okay, so here's Lisa Cordeiro.
So as David was describing about the overlap between autism and Fragile X, I want to talk a little bit more about one of the core features of Fragile X, which is anxiety, particularly social anxiety, and how that overlaps with autistic symptoms. Okay. So clinically, we see a lot of overlap in the behaviors associated with autism and a lot of behaviors that are associated with anxiety. Things like gaze avoidance, withdrawing from social situations could be considered characteristic of autism, but is also seen in social phobia, which is an anxiety disorder. We also see a lot of rigidity and problems with changes in routine and problems with being flexible to changes in the environment in autism, and also in anxiety and things like OCD, which is another anxiety disorder. And because anxiety, and particularly social anxiety, is so important in Fragile X, it's an interesting model to better understand the overlap of autism and anxiety. So the literature on anxiety in idiopathic autism, that is autism without Fragile X syndrome, is somewhat limited. And most of the research hasn't used good clinical interviews to assess the anxiety disorders. Of one of the few studies that did use clinical criteria, 84% of the children with autism were found to have at least one anxiety disorder. And most of the other studies of anxiety and idiopathic autism have been done in high-functioning autism or Asperger's, and they found incident rates of about 22%, and scored significantly higher on anxiety compared to a group of controls without autism but had developmental delay, as well as to typical controls. And as far as the research on anxiety and Fragile X, Dr. Hessl talked about it being part of the behavioral phenotype, and it's been pretty well documented around rates of about 40% using parent report questionnaires, that boys and girls with Fragile X experience clinical levels of anxiety, things like shyness, fearful behavior, withdrawing from social situations. And in one of the few studies that actually used diagnostic criteria for anxiety disorder, they found 65% of females with Fragile X to meet criteria for avoidant personality disorder, which is basically social avoidance. So the literature is limited in both anxiety and Fragile X and anxiety and autism, and so to that end, in our lab, we've been trying to collect better information about anxiety. And so far, among the males with Fragile X syndrome that have participated in the research, we find really high rates of specific and social phobia. And I want to point out that the dark blue region represents those who have met full criteria for the disorder, and the light blue region represents those who would meet criteria if some initial screening questions were answered. And those initial screening questions usually require some type of verbalization from the child or reporting from the child about their anxiety symptoms, so things like I worry a lot in a social situation, or I'm scared to be around people. And for a large majority of, particularly the males with Fragile X with an intellectual disability, that's difficult for them to report. So I just want to point out that if we were to take out that requirement of those screening questions and them having to say they're anxious, a great many more would even meet criteria for these anxiety disorders. It's also interesting that a large portion, about almost 30%, meet criteria for selective mutism, which is not that very common in the population, and that's a complete refusal to speak in social situations. And among females with Fragile X syndrome who have participated in the research, we see a similar pattern. There's still a lot of anxiety disorders, but in a smaller percentage of the girls. One marked difference that you can see is over 70% of our sample have met criteria for post-traumatic stress disorder, or PTSD. And we don't quite understand the reasons behind all of that. A lot of people might first think that this means there's a lot of sexual or some other type of abuse going on. But actually, a lot of these girls, the PTSD is in response to any traumatic event. It could have been a hospitalization or a very bad situation at school, and the situation has lasted, the symptoms are still affecting them. And so the next steps with the study, it's still going on, so if you'd like to participate, please let me know, is to look at how exactly the autism symptomology in Fragile X is relating to these anxiety disorders we're seeing in Fragile X. So now we've better documented the types of anxiety and the severity in Fragile X syndrome. We'd like to see how this contributes maybe to the autistic symptoms we also see in Fragile X. And so with that, I'll hand it back to David to talk more about those connections.
So before I move on to the next few topics, I just wanted to follow up with something related to what Lisa was talking about, which is that the study is ongoing, and you might have noticed that she only presented data from individuals with Fragile X syndrome, but in fact the study also includes children with Down syndrome. So that's a group that we want to use as a comparison. Very important population for us to look at, and it also includes typically developing kids. So we welcome any parents who would like to enroll their kids in our research, and you can contact either Lisa or myself about that. So one thing that we've done as a research community is try to look much more carefully into the reasons why people with autism and related disorders have so much difficulty giving eye contact. So as you know, that's one of the symptoms that's part of the reciprocal social interaction deficits that are part of the diagnostic criteria. So some prior studies that have been done are really fascinating. Ami Klin and his group at Yale showed a movie called Who's Afraid of Virginia Woolf, or at least part of it, to a group of individuals with, I think they were adults, with autism, and a comparison group, and they found that individuals without autism with normal development very much looked at the eye region of faces to get information about the social and emotional interactions that were happening during these clips. Whereas people with autism tended not to look at the eye region, but more often to the mouth or to other non-social aspects of the environment or the background. Following that, there was an interesting study done by Richie Davidson's group in Wisconsin, and they actually combined functional MRI with, or brain MRI with information about where individuals were looking on pictures of human faces. And what they found is that when individuals looked more to the eye region of human faces, they had stronger activation of a region of the brain called the amygdala, which is involved in emotional appraisal, and is, a lot of studies have been done showing that it's linked to processing fear or threat. So these studies really prompted us to consider how these mechanisms might play out in Fragile X, which is another disorder where eye contact is such a big problem. So Faraz Farzan, who is a graduate student at UC Davis, works with Susan Rivera. She and I developed a protocol to address this by using an infrared eye tracker in the lab. And this is a device that can determine where you're looking, either on a computer screen, or there's even another version of it where you can determine where somebody is looking in a natural environment, like in a naturalistic social interaction. But in our case, we just used stimuli shown on a computer screen. And what we did is we showed participants in the study 60 different photographs. And these were photographs of human faces that either showed calm or kind of neutral, happy, or fearful facial expressions. And each of those 60 faces were preceded by an image, which I'll show you in a second, that is basically a scrambled version of the face. So I think I can move ahead and let me say a minute also about the participants. So this was a pilot study. We just had 16 individuals with the Fragile X full mutation, and then 16 age-matched controls. So the task went like this. Within each sort of stimuli set, participants saw a gray background screen, and then they saw for 1 second a scrambled image of a face. And that scrambled image was the scramble of the subsequent face. And the reason we did that is because we were not only interested in where on the face individuals with Fragile X were looking, but also how their pupil size was reacting to the image. And the reason we were interested in that is that you might know that pupil size can change depending on how much light is coming into the eye, but also it can change depending on your emotional response to whatever you're looking at. And so that emotional response is driven by the sympathetic nervous system, that fight or flight part of the central nervous system. And then there's just another trial example. So now I'm going to play you a movie that shows in real time where a participant with Fragile X was looking when they watched this part of this protocol. Okay, so what you saw there is quite a bit of focus on the nose and the mouth region, and a few kind of darts over to the eye region, but that were not very persistent. So this is just some examples of how the data kind of looks with two different participants, one with typical development and one with Fragile X, and you can see these are points of fixation that are, the bigger they are, the longer time a person spent looking at that particular place. So these are just kind of representative pictures of that eye scan pattern. So we measured percent looking time, or the proportion of time that individuals with Fragile X and typical controls looked at different regions on these faces. And what we found was pretty much what was expected based on what we know about the phenotype of Fragile X, which is that they looked much less often at the eye region than individuals with typical development, and they looked more at the nose region. Something that we didn't necessarily predict, but they had to look somewhere else besides the eyes, and we weren't sure what we were going to find. And so there's some interesting hypotheses that we have about why that might be. If there's time, we could talk about that. For happy faces, same pattern, basically. And for fearful faces, slightly different. There was no significant difference for the nose region. The eye region difference held up, and then for whatever reason, they tended to look more often than controls at other parts of the image. Now, in terms of change in pupil diameter, this is where I think probably the more novel part of the study that's, I think, the most interesting. So this is time going across here. The picture comes on at this point, and then these lines are telling you how the pupil diameter is changing across time within each group. And you can see here for calm faces, there's no significant difference, although the group with Fragile X is somewhat higher. But we saw significant differences for both of the emotional types of faces, with the Fragile X group showing a greater pupil dilation to both the happy faces and the fearful faces. So here we have some evidence that possibly one reason that individuals with Fragile X are looking less to the eye region is because there's an emotional, possibly aversive emotional component to looking at that kind of social stimuli. So the new study that we're doing, that Paula Weinstein-Waddell, who's in the audience, is doing with me, is very exciting because we're actually expanding it now to look at how individuals with Fragile X and autism and controls process visually images that are not static, but actually more realistic social stimuli. And so for that study, we're enrolling individuals with Fragile X, with autism and without autism, a group of individuals with Down syndrome, and also individuals who have autism not due to Fragile X. So now I'm going to show you an example of one of the clips that's used in this protocol. And Paula's done a great job in recruiting some pretty talented actors to give us these performances. So I'm going to show you an example.
My birthday was yesterday. I had a great time because my friends all came over to visit me. Do you like it when your friends come and visit you?
So the idea here is to, first of all, there's different actors, and each actor shows a different emotion or portrays a different situation that elicits their emotional reaction. And we also tried to make it as engaging as possible. And although it's a little bit contrived, the idea is that the participant is asked to try to respond to the questions so that they feel like it's a little bit more like a real interaction. So we've literally just started this study, but I just wanted to show you kind of how the data looks just from the first participant that we've seen, a young girl with both Fragile X and autism. And you can see here, both with this hotspot representation, and then also this information down here about sort of fixation, where they're fixating and for how long, that at least in this case, this individual is looking very much at the mouth and not really at all at the eyes. In fact, in all of the clips, I can't remember, Paula, how many clips there are? 20? She actually never looked at the eyes for the entire protocol. So, and that fits very much with her clinical presentation. Just, we all noticed right away that she had almost no ability to give eye contact. Another collaboration I have going is with Susan Rivera, who's doing functional imaging with us. And what we're doing in those protocols is real exciting because we're actually combining some of these eye tracking technologies with functional brain measures. And this is just an example of one of the tasks where people basically look at two faces on the top showing different emotions, and then a face on the bottom, and they have to decide which face on the top matches the emotion of the face on the bottom. And that's another amygdala activation task. And what we've done here is superimposed all of the eye scans for not just this picture, but all of the other trials sort of on top of each other. And the reason that we wanted to include eye tracking is because in our prior studies, there was a lot of concern about how activation might be different depending on whether the people are actually looking at the faces. So if individuals who have these conditions are averting their gaze and not looking at the eyes or not looking at the faces as much, then that could bring their activation level down. So this is a way where we can actually control for that. Okay, so now you get to participate. This is a task that we're using in the lab that was developed by Simon Baron-Cohen, and it's called the reading emotion in the eyes task. And what you do in this particular test is you look at a picture of a person's face and it only shows the eye region, and you have to decide which of these either thoughts or emotions best describes what that person is experiencing. So maybe we could just have a show of hands. Who says playful? Okay. Comforting? Okay. Irritated? Bored? Okay, so, sorry Claudia. You missed that one. Yeah.
Do people with Fragile X have normal hearing?
Normal hearing?
Yeah. Because if you're lip reading, what if they're actually trying to interpret?
Yeah. In most cases they do, but I know that Rondi has mentioned that you know, like ear infections are really, really common. I think probably more common than the general population. So they tend to have tubes placed more often. We've not had many parents report like clinically significant hearing loss though. But I agree. I mean, that's something that we could definitely want to check into.
I mean, that
for sure.
Come for all the other sort of social, aberrations, like looking away necessarily, but you wonder about the focus on the mouth.
Right. Or maybe even if they hear fine, they may still need to get more information from the mouth to understand what's going on. Yeah. Okay, so that's one example of a test that we do. Oh, the answer. Sorry. Yeah, most of you were right. It's playful. And you should keep in mind, though, that there's no like absolutely correct answer. So the way that this test was constructed is they gave all these items to a large group of people who are normally developing, and they had them all, rate their answers. So there's a certain percentage of people who say playful, and then smaller percentages who say these other things. So if you said playful, you're kind of mostly right, but there's no like absolute correct, if you know what I mean. So the other task we're using is called the Movie for the Assessment of Social Cognition. And this was developed in Germany. And so you'll have to forgive that there's an English voiceover. And it's done pretty well, but it's not great. But the way that this task works is that it's also done on a computer. And you're introduced to 4 different characters. And what you're going to be doing is watching a movie that's interrupted every so often. And you're asked questions about what the characters are feeling or thinking during this, I think it's like a dinner party scene. Let's see, so this gives you just sort of the instructions, including, if you're not exactly sure, you're supposed to guess, so that we always get an answer to all their questions. So these are all the characters and their names, and then I think the next slide is going to be the first clip. So what you want to do is pay attention to this clip, and then there'll be some questions for you to answer.
Hello.
Hi.
Hey, you. Surprise? I thought this was where you live.
So how are you?
I'm fine. But take a look at you. You look terrific. New hairstyle?
Yeah.
Classy.
Thank you.
Okay. So the question is, what is Sandra feeling at the moment that the clip stops? There she is, so you can remember who she is. So here's your options. Okay, we'll do hands again. Who says A? B? C? Okay, D? Okay, so D is mostly correct. That's what the normative data said. So I think that they're both, somewhat correct, but that's the answer, supposedly. So there's just a number of these clips, and then a person gets a total score. If any of you are around tomorrow morning, Susan Rivera is going to be talking about this brain imaging research that we're doing collaboratively, and she's going to be talking about how individuals' performance on this test correlates with their brain activation in MRI. So it's real interesting data that we're starting to pull together. Another area that I mentioned before that there's a lot of overlap between autism and Fragile X is in the sensory domain. And we know both clinically and from a number of studies that there are a lot of sensory abnormalities in individuals with idiopathic autism, both hypo and hypersensitivities. And you can see some of the examples in each of these sensory domains. So before I even got interested in this topic, there was a study of something called sensory motor gating in individuals with Fragile X. And what was found, this sensory motor gating is basically how well the nervous system can filter out sensory information so that we can focus on what we want to focus on. And so what this study showed was that individuals with Fragile X had very substantial deficits in this measure of sensory motor gating. And one of the real interesting things about the study was that a measure of autism was very strongly correlated with the degree of this sensory motor deficit. So that prompted us to carry on with this type of work. And we initiated a Prepulse inhibition study in Fragile X. Prepulse inhibition is just one way that you can measure sensory motor gating. We felt that there was a real need because of clinical trials that were being developed to find feasible and objective and reproducible measures that could be used to track treatment response. We were looking for measures that would also be reflective of Fragile X deficits, that they could be used in both animal studies and in human studies. And then I'll be talking more about this in a couple minutes, but also measures that would be mediated by the metabotropic glutamate receptor system. And that's a system that's been very strongly implicated in the pathogenesis of Fragile X. So it just so happens that PPI really satisfies all of these. So the way we do this work is we apply 2 electrodes under the eye, and these electrodes measure the magnitude of the eye blink startle response. So you know when you hear a sudden sound, you'll kind of blink or have a startle reflex, and we can measure how strong that is from those 2 electrodes. So the measure of prepulse inhibition is determined by basically a comparison of how strong your response is when you hear a startle, like a loud sudden sound all by itself, in comparison to when you hear the same sound, but when it's preceded by a softer tone. And when there's a softer tone, it's sort of like the nervous system's warning that you kind of want to dampen your response. It's an automated thing, it's not a conscious thing, but normally you see a much reduced startle reflex. And so we can measure the percent of inhibition that we get from here to here. So in this study, what we found, which was basically replicating that other smaller project that I mentioned earlier, is that individuals with Fragile X, both males and females, had very substantial impairment in prepulse inhibition. So this is kind of just replicating what had been shown in the other study. But what was really, I think, the important finding was that we had really good test-retest reliability, meaning that when one person came in and had the test done the first time, then they came back a couple weeks later and we redid the test, they wound up having very similar prepulse inhibition values. And that's important because if you want to use something like this as an outcome measure in treatment studies, you want it to be stable when there's no change in intervention. So we were real happy about that. And the nice thing is that that led to our using it in a psychopharmacological trial, which is the first trial of a mGluR5 antagonist in people with Fragile X syndrome. So the theory is that when this protein, FMRP, is absent or when it's deficient, there's no break on this synaptic connections, no break on this system. So this system kind of runs out of control, and synaptic connections are weaker, and the dendritic spines have unusual shapes and so on. In the animal studies, these abnormalities, both the structural abnormalities and the behavioral abnormalities in the animal models are normalized when the animals receive an mGluR5 antagonist. So there's a lot of animal literature now supporting this. So this is the first trial in humans, which was just came out in Journal of Medical Genetics. And what we found, and I should say it was just really a study mainly looking to find out whether there were any adverse reactions. That was the main purpose. There was only a single dose. We didn't find any adverse reactions. But interestingly, 6 out of the 12 people who participated in the study had a really nice improvement in this measure of sensory motor gating. So this just basically, it's not blinded and it's not placebo-controlled, but it encourages us to do that. And some of these more carefully controlled, bigger studies are going on actually right now. Another interesting parallel between Fragile X and idiopathic autism is findings that are pertaining to the serotonin system. So as you know, or many of you might know, the serotonin is one of the neurotransmitters that's really involved in regulating our mood. And it's been implicated in depression and anxiety disorders. And so there have been a number of studies, including this one, showing that these genes that regulate transportation of serotonin in the synapse determine severity of autistic symptoms. And so an interesting finding that we had when we explored this in our population of individuals with Fragile X was that this same gene moderated the amount of aggression that males with Fragile X showed, and it also was a moderator of the amount of stereotypy that they showed. So it seemed to affect both their aggression, which can be also associated with irritability, but also these other symptoms like hand flapping and other kinds of stereotypies. So there was some similarities between these different studies. And in most cases, the studies that have looked at serotonin transporter measures in autism have suggested that when there's an increase in that activity, there's more significant symptoms, which is basically what we found here. Another way that I think we can make some generalizations from Fragile X to autism is in the cognitive domain. And what we found in a recent study that was a collaboration between the Mind Institute, the Fragile X Center here at the Mind Institute, and also the Fragile X group at Stanford, where I came from before here, we basically collected IQ test data from about, 217 individuals with Fragile X syndrome using the Wechsler Intelligence Scale. And what we learned from this analysis was that an unbelievably high percentage of people with Fragile X scored at the very bottom of this test. They had a floor effect, which means that they obtained the lowest possible score that you can possibly get on this test. And these numbers, this is overall on the left, and then males and females, shows you the percentage of people out of the 217 that got this lowest possible score. And so it's really remarkable that between 60 and basically 90% of males hit the floor of this test. And you should recognize that these are tests that are just regularly given throughout the United States and elsewhere to individuals with intellectual disabilities, just like these guys with Fragile X. So it's extremely likely, if not definite, that this problem exists in lower functioning people with autism, Down syndrome. I mean, you could name many different conditions where this would be an issue. So what we did was we thought, how can we make use of this data in a different way? So we wrote to the publishers of this test and we asked them for the descriptive statistics from their standardization study. So when they, develop the test, they give it to hundreds and hundreds of kids with normal development, and they use that to construct the norms for the test. So they, because it was for research, they released those data to us, and we were able to reconstruct a new normalized score for all of these kids with Fragile X that tells us basically how much do they deviate from normal. And so what we found is that there were no floor effects at all with this new measure. And when we correlated this new measure with another measure of functioning called the Vineland, which is a measure of adaptive behavior like daily living skills, socialization, things like that, what we found is that when we did that correlation with the usual standardized score that had all these floor effects, we found very weak correlations with all these subjects piling up on the bottom. These are just two examples from that IQ test. But when we used the new normalized score, we had a very much stronger correlation, which basically proves the point that there is variability in these tests that is not being represented when you, report the scores in their usual way when you use the standardized scores from the manual. So this method is not really available yet, unfortunately, for people to use in clinics and things. It's really just for research, but it's our hope that with some more replication and publication of this, that there might be ways that this kind of method could be used clinically. Okay, so my last topic is about aging. So a number of years ago, there was a paper that was published suggesting that children with autism had higher levels of amyloid precursor protein. This is the protein that confers more risk for the plaques and tangles that you see in Alzheimer's disease. But it so happened that they had 2 people with Fragile X in their sample of autism. And they said here that inspection of the data shows that the individuals with Fragile X had the highest levels of secreted beta amyloid precursor protein, and they made this kind of conclusion that it could be the Fragile X, but not the overall group that might contribute to this elevated APP. So lo and behold, a few years later, a study came out that actually showed that FMRP, which is that protein that I mentioned before, when it's absent causes Fragile X syndrome, actually mediates the interaction between the mGluR5 system and amyloid precursor protein. So basically the simple way of saying it is when FMRP is deficient, APP goes up. So our hypothesis that we're now hopefully going to explore in a new study that we've proposed is that individuals with Fragile X and autism are at increased risk for Alzheimer's type dementia as they age. So why would it be, other than that, why would it be important to study aging in autism and Fragile X? So one question that's been posed in some of the scientific literature and also just on the internet is, if autism is as common as we think, where are all the old people with it? And I think there's a lot of people wondering that. The new figures suggest that 1 in 150 people have an autism spectrum disorder, and yet they all seem to be kids. Either that or all the adults are somewhere in hiding or have compensated somehow. If they are out there, how do we find them? The reality is that because autism was, identified and described not that long ago, many of these very elderly people probably have diagnoses from an earlier period before autism was even recognized or diagnosed. So that's one problem. So a reason that it's important to do these studies is because we need to know how these people with autism and Fragile X age, both from a medical, psychological, and a service delivery perspective. We really just have no idea what their needs are and how they're functioning. Another issue or complication is once they're identified, how do we make a diagnosis? Most of the tools that we have for diagnosing autism are all developed for toddlers and school-age kids and young adults. We don't really have anything that's validated yet to diagnose autism in an elderly person. So what we think is that Fragile X offers a really powerful opportunity to study aging in autism. That's because we know we can find the people. All we have to do is look at our pedigrees of families who are affected by Fragile X, make contact with the, usually one of the moms or parents in the family, and just work our way back in those family trees and ask about those more elderly family members. The ones who have mental retardation are probably have Fragile X and have somewhere between 30% and 50% chance of having autism. So this is a way that we can at least get started addressing some of these questions. There have been no published studies and no grant support to study aging people with autism. NIH, that I could find in their database, has never funded a study of elderly people with autism, and there's no published information at all in the literature that we could find. I think we talked about this. And then Dr. Hagerman, who you all probably have heard about, she came back from I think it was an intellectual disability meeting recently where there was a study presented where they showed that FMRP, not in people with Fragile X, but normally is involved in adult neurogenesis. So normally FMRP plays a role in the nervous system and how it develops even in mature individuals. So just to go back to the original question, what do we learn about autism by studying Fragile X? So as I mentioned at the beginning, it's a clear example of an autism subgroup. There's been lots of efforts here and other places to phenotype autism, to find identifiable subgroups, and we know already that this is one of them. And because we already have identified this subgroup, we can test models for understanding the etiology and treatment in a more homogeneous condition like Fragile X, and then once we know what those models are, we can test them out in more heterogeneous groups like idiopathic autism. We can study the brain mechanisms underlying particular autistic behaviors within Fragile X and then see if they also play out in regular autism. We can study how background genetic and environmental factors play a role in the expression of symptoms. And that's because if we have a more homogeneous group, we have a better chance of seeing what those factors are rather than having a much more mixed group of individuals with autism. And finally, we can learn how to investigate and test novel therapeutics like these mGLUR5 antagonists through animal and human studies, because we have both an animal model and a human condition. So I have tons of people to thank. I don't know a better team in science. I mean, it's just the most collaborative group that there is, and I won't go through everybody here, but Rondi is sort of my primary mentor and my inspiration with all of her wonderful ideas that I have to kind of keep up with and try to help answer. So I have a lot of thanks to her, and Lisa especially for coordinating the lab and keeping everything moving forward and not sinking, basically. And then many other members of the team, which we call our Fragile X team, and then of course all the participants and their families for donating their time. So thanks, I can take some questions.
Do aged Fragile X patients show neuronal loss?
Show?
Neuronal loss.
Neuronal loss. I know from the few MRI scans that are available that there is atrophy, but it's not clear at all because there haven't been group studies whether that atrophy is more than age expectations. Claudia maybe knows more than me. Do you know anything about that, Claudia?
No, the studies, there are not very many studies done on Fragile X brains because people they're hard to come by.
Right.
And we're just starting to study a few right now. We don't have the two that we have. We don't have definitive stains back to
would you say that there's significant atrophy?
No.
Or not really?
No.
Okay.
Are these studies measuring the expression of the FMR1 gene in patients with idiopathic autism to show if there is any detectable variance due to polymorphisms other than the CGG repeats? If the gene is with normal number of CGG repeats, still there may be some polymorphism which influence the level of expression, which may be a predisposing factor for idiopathic autism.
Yeah,
I guess the easy way to answer that is to study the level of FMR1 expression in patients with idiopathic autism, or just do association study in a large group of idiopathic autism kids for association with particular polymorphisms around the FMR1 gene. Are there any studies?
there's nothing like that, and I think it's a great idea. I know that I don't know if this is related, but in some of the kind of preliminary studies that we've done, like the imaging studies that I mentioned with Susan, we're finding that there are some associations between FMR1 measures and either brain measures or clinical measures in the control group. So we don't know if that's just some chance thing that's not real, or if there's some real effect of slight variations in FMR1 within the normal population. So it could also apply then to
I think it would be
autism.
I think it would be reasonable to expect, particularly given recent report where MECP2 gene, which is responsible for Rett, was found actually that there is a polymorphism in one of the introns, which is strongly associated with idiopathic autism, not with Rett syndrome.
Mm-hmm.
So obviously the gene has something to do with not only with Rett, but with idiopathic autism.
Yeah, it sounds very good to me. Other questions? Okay, thank you.
The UC Davis Mind Institute began in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, learning disabilities, and other brain disorders is helping children achieve their fullest potential. Please call or visit our website to find out more about current studies, our research team, and upcoming events.